So this is our fourth class on Bacteriology Unit 1 Cell Organization of SEM 2. And now when I look into the structure external to cell wall, the first thing which comes to my mind is glycocalyx. What is this glycocalyx? Glycocalyx is nothing but the term which is used to refer to network of polysaccharide substances found external to cell wall. What is glycocalyx? Glycocalyx is nothing but glycocalyx is a network of polysaccharide substances, which is the currently accepted term which is used to refer network of polysaccharide substances found external to the cell wall. It can be from the thickest capsule to the thinnest slime layers. Okay, they are sticky and viscous. So they are basically sticky and viscous. They can stick and are viscous in nature. Viscous means sticky only. They are gelatinous polymer. They are composed of polysaccharide, polypeptide, okay, or both. They can be composed of polysaccharide and polypeptide. You can remember this. They can be composed of polysaccharide or polypeptide. They are actively synthesized inside the cell and released outside. So these there can be a question then, where are glycocalyx synthesized, inside or outside? They are synthesized inside and then released outside the cell. Now we have glycocalyx. This glycocalyx can be of two types. First is capsule and second is slime layer. So we are going to see first capsule. What is capsule? So when we talk about capsule, it is a protective layer outside the cell wall which is thick, that is well organized in a tight matrix and not easily washed off. Then this is known as capsule layer. So this capsule layer is not a weak layer. It is a very strong layer with a tight matrix, tight binding, and made it is thick layer, not a thin layer even. It is a very thick layer, and whenever it is not washed off easily, this is known as capsule layer. Now capsule is produced by some bacteria, not all species of the bacteria. So this capsule is not produced by all bacteria or all species of bacteria, but by some. Composition: capsule is made up of a complex polysaccharide held in loose gel. It is made up of a complex polysaccharide held in a loose gel, which is made up of glucose. So it is made up of a complex polysaccharide that is kept in a loose gel, which is made up of again glucose, rhamnok, uronic acid of various sugars, 2-keto 3-deoxyglucouronic acid, and other organic acids also like pyruvic acid, acetic acid, etc. Some bacillobacteria, such as Bacillus anthracis, Bacillus anthracis, which is causative agent for anthrax, has a proteinaceous capsule composed of poly-D-glutamic acid. So it can also have poly-D-glutamic acid. Now B anthracis do not produce capsule when it grows outside the organism. When this B anthracis grows outside the organism, it do not produce capsule, but it produces capsule when it infects an organism. So when it infects an organism, then it ensures that it produces a capsule, but when it is outside the organism, it is growing outside the organism, it does not produce capsule. So what you understand from this, glycocalyx is a term used to refer to the network of polysaccharide substances found external to the cell wall. So these are the polysaccharide substances. It can also be made up of polypeptide. They're actively synthesized inside the cell and released outside. So glycocalyx is prokaryotes have variety of structures outside the cell wall, okay, and that can function in protection, attachment of objects, and cell movement. And among this one is glycocalyx. Glycocalyx is currently accepted term which is used to refer to the network of polysaccharide containing substances but external to the cell wall, from the thickest capsule to the thinnest slime layer. The bacterial glycocalyx is viscous, sticky, gelatinous polymer and composed of polysaccharide, polypeptide, or both. Its chemical composition varies widely with the species. For the most part, it is made inside the cell and secreted to the cell surface. Glycocalyx can be of two types. One was our capsule, which we studied, and another is our slime layer. Now we'll look to the functions of the capsule. What are the functions of the capsule, and what are they? Okay, let's see. Now let us look into the functions of capsule. What are the functions of capsule? First is, capsule is, because it is viscous and sticky, so it helps in the attachment of bacterial cells to various surfaces such as plant root surface, teeth, human teeth, and tissue because of its sticky nature. So first point I'm trying to explain to you, it helps in the attachment of bacterial cells to various surfaces like the plant root surface, teeth, tissues. One example, if you take Streptococcus mutans, it causes dental caries. What is dental caries? Caries, that is our dental cavity basically. It binds tightly to the human teeth like a cemented material by the help of capsule. This dental carie happens because Streptococcus mutans bind to your teeth like a cemented material because of its capsule. So this is the first very function of capsule. Second is, capsule helps in identifying disease causing ability of microorganism. Example, Streptococcus pneumoniae. So it helps to identify the disease causing. Like if you take an example of R2 of Streptococcus pneumoniae R2 strain, it is non-virulent, so it does not have capsule. S3, it is virulent, so it contains capsule. So the Streptococcus pneumoniae can be identified like, if you have a virulent, then you can say that it is having capsule. If it is non-virulent, it is not having capsule, so it is S3 and R2. You can identify. Third point, the capsule resists phagocytosis caused by host phagocytes. So it reduces the phagocytosis caused by the host phagocytes. So capsule is antiphagocytic in nature. Capsule is also hygroscopic in nature. So it has the decent amount of water storing capacity. So next point is this, capsule protects bacterial cells from desiccation, bursting because capsules are hygroscopic. Hygroscopic means it prevent desiccation and can store a significant amount of water. So it can store a significant amount of water and protect the cell from desiccation. Capsules are used by bacteria such as S. mutans, Streptococcus mutans. This was only causing dental caries. This also uses the capsule for utilizing energy by breaking the capsule and utilizing sugar during nutrient deprivation. So what S mutant do at the time of nutrient deprivation? They break the capsule, utilize energy from it by sugars which it get from breaking. Next, let us look. Next is, capsules prevent the attachment with bacteriophages and various hydrophobic toxic materials such as detergent. So it protects from the toxic materials such as detergent. How it works? It blocks the attachment of bacteriophages. How? Making the attachment site of the bacteriophages. Okay, masking. It is masking the attachment site of the bacteriophages. Next point is, it helps to form biofilm. What is biofilm? Biofilm is the aggregation of different bacterial species with association of fungi. It is mutualistic or commensal. So this is our point, that capsules help to form biofilm in many bacteria, bind with solid surface to form thick layer of cells called biofilm. And now the last point, which I have not written, the eighth point I am writing over here. The eighth point is very important. It is what? If the capsular material contains uronic acid. What? If the capsular material. Okay, so if the capsular material contain uronic acid, then it promotes stability in bacterial suspension and prevent aggregation. Okay, so this was our last point and very important point. So if the capsular material is having uronic acid, then it promotes stability, and this is a very important point. You can mark it as a star mark. Next, we come to our second glycocalyx type, that is slime layer. Slime layer is opposite of this. Capsule was thick, well organized, tight matrix. It is opposite. Slime layer is thinner, less tightly bound, unorganized, and easily washed off. It is usually composed of polysaccharide. It was having polysaccharide and also some amount of polypeptide, right? But this is composed of polysaccharide mostly. Even bacteria has at least a thin slime layer. Even the bacteria, every bacteria, sorry, not even, every bacteria has at least a thin slime layer. Now capsule was not having all, not contained by all bacteria, but every bacteria will have a slime layer, at least a thin slime layer. Now we have function. Slime layer also provide adherence, same adherence. It allows to take up nutrient in the near cell. From the near cell, it allows to take nutrient. It protects the cell from drying. It helps the cell to remain wet. It has a gliding bacteria often produce slime. Okay, gliding bacteria in some cases have shown motility. It helps in motilities also. So this was all about slime layer. The last thing which I thought of giving you is the difference between capsule and slime layer. Capsule is thick, slime layer is thin. It is well organized, it is unorganized, it is tightly bound, it is loosely bound. Not washed off easily because it is strong, tight matrix, washed off easily. Okay, can visualize under microscope because it is well organized and tightly bound. Matrix, it cannot be visualized. So this is what I have taught you today. This was your fourth class. You can take a screenshot of it and study it.